Diagnostic genetic tests give you a definite yes-no answer about whether or not your baby has a chromosomal or genetic condition. Most prenatal diagnostic tests are done on samples collected after carrying out an invasive procedure. The two most common invasive procedures are chorionic villus sampling, CVS. CVS is usually done between 11 and 14 weeks of pregnancy. A long, thin needle is put through the tummy, sometimes through the vagina, to collect a tiny piece of tissue from the placenta. This tissue sample is then tested in a laboratory. Amniocentesis, amnio. Amnio can be done after 15 weeks of pregnancy. A long, thin needle is put through the tummy to collect a few teaspoons of the liquid around the baby, amniotic fluid. This fluid is then tested in a laboratory. CVS and amniocentesis carry a small risk, approximately 0.5% or 1 in 200, of causing miscarriage, which means the decision about whether to have them can be difficult. Unfortunately, in most cases, there is no other way of knowing for sure whether your baby has a chromosomal or genetic condition. There are 3 different tests that can be carried out on a sample from CVS or amnio. A rapid testing technique, called quantitative fluorescent polymerase chain reaction, or QF-PCR for short, is used to get a result quickly for certain chromosomal conditions. It uses the sample from the CVS or amnio and looks for genetic markers that indicate chromosomes 21, 18 and 13. If there are too many of these markers, it shows that the baby has either Down syndrome, Edwards syndrome, or Patau syndrome. It can also sometimes detect conditions associated with the sex chromosomes, such as Turner's syndrome. The result is usually available within 3 days. Another type of test is called a karyotype. This takes longer than QF-PCR because before analysis, cells are grown in the laboratory in order to separate out and identify all the baby's 22 pairs of chromosomes. Once the chromosomes have been identified, they are arranged in pairs in numbered order from 1 to 22, plus the 2 sex chromosomes, and a special dye is used to highlight regions, or bands, of each chromosome. Then each chromosome is examined under a microscope to check for any changes that may be significant. For example, extra or missing chromosomes, or parts of chromosomes that have changed position. It usually takes from 10 days to 2 weeks to get the results. In recent years, a different test has started to replace the karyotype. This is called chromosomal microarray analysis, CMA. CMA can examine a baby's chromosomes much more closely than a karyotype. The laboratory technique compares a baby's DNA from the CVS or amnio sample to a reference typical DNA to check for changes. CMA can detect extra chromosomes, as in Down syndrome, or missing or additional parts of chromosomes, but can go further to find very small changes in the DNA known as copy number variants, CNVs, where a number of genes are missing, microdeletion, or when there are extra genes, microduplication. Sometimes these changes will explain what has been seen in the baby on scan, but not always, and sometimes the effect of the CNV can be difficult or impossible to predict. It usually takes from 10 days to 2 weeks to get the results. The most recent development in prenatal genetic testing is often called next-generation sequencing. This refers to prenatal whole genome and exome sequencing. Sequencing the whole fetal genome, reading all the letters in the baby's DNA, means an extraordinary amount of information is generated, some of which will be very difficult and time-consuming to interpret. Researchers have found that sequencing the part of the fetal genome called the exome may be more practical and beneficial. The exome makes up no more than 2% of the whole genome, but contains the majority, around 85%, of the genes that we know cause genetic disorders. Sometimes the laboratory can use a gene panel in the sequencing. This means they will only test for genetic changes that are known from existing evidence to affect the baby's health or development. This is a new technology and is just starting to be offered in the NHS. Most experts recommend that it should only be offered when all other tests have failed to give an explanation for unusual scan findings. It does not replace chromosomal microarray because whole exome sequencing does not reliably detect genetic copy number variants. It takes about 3 weeks to get a result. It is important to know that the whole genome or exome sequencing will not always find out why a baby is not developing as expected. Occasionally, the testing will reveal unexpected genetic information about the mother's health. While all the tests mentioned so far can only be done after a CVS or amnio, some genetic conditions can be diagnosed from a simple blood test from the mother. A cell-free DNA testing technique, known as non-invasive prenatal diagnosis, NIPD, can now diagnose some single gene disorders through a maternal blood test. A single gene disorder is a genetic condition caused by a change in one particular gene. Examples of single gene disorders are sickle cell disease and cystic fibrosis. There are some inherited genetic conditions that can currently be diagnosed by NIPD in NHS laboratories in the UK. If you know you carry a single gene disorder, you should speak to a genetic counsellor to see whether NIPD might be available. NIPD can also be used to find out the sex of the baby as early as 9 weeks. This can be helpful if expectant parents know they carry a sex-linked condition, usually a genetic condition that only affects boys, such as Duchenne's muscular dystrophy.